Last but not least, it is my pleasure to have our very own Dr. Dorothy Teegarden, Professor of Nutrition Sciences here at Purdue and Director of the WGHI, and one of the 2023 Catherine P.G. Grant awardees, talk about the role of the proteome of cycloplastic lipid droplets in metastasis.
Okay, I do walk around, so I can at least walk. So today I'm going to talk a little bit about proteomics as well. And so we are particularly interested in our laboratory about breast cancer metastasis. And the reason why is if you look at if a breast cancer is diagnosed and it's still localized, the 5-year survival rate is really quite good. However, upon diagnosis, if it has metastasized to a distant site, the 5-year survival rate drops dramatically. And so it's really important for us to understand how metastasis occurs, what drives it, and also metastasis accounts for greater than 90% of all cancer deaths, not just breast cancer, approximately 700,000 women die each year worldwide from breast cancer. So it's a critical public health problem for us to identify how metastasis occurs. So I'm going to talk about lipids and fats in cancer. And intriguingly, we've known about lipid accumulation or fat accumulation in invasive breast cancer since the 1970s. And it's been identified that this accumulation, lipid droplet accumulation or fat accumulation, occurs in many different kinds of cancer, including breast cancer. And after 50 years, we still don't know why or what about this lipid accumulation promotes metastasis. It's thought maybe it provides energy because fats provide energy, but we really don't know. And so this lipid accumulation in metastatic cells in particular is very important because it's associated with more aggressive metastasis and poorer patient outcomes or mortality. So it's really important for us to understand what happens with this lipid accumulation in metastatic cells. So I'm going to talk, what is this cytoplasmic lipid droplets? How are fats stored? So this is actually a micrograph, an electron micrograph from our lab in a non-metastatic cell. And here's the nucleus, and so it's just a part of a cell. And these are our lipid droplets, or where fat is stored in the cells. If you look at the metastatic cells, you can see these big lipid droplets that occur in metastatic cells, which is consistent with what I just told you, that more aggressive cells have these big lipid droplets. And so what a lipid droplet looks like, here's a cartoon of a lipid droplet. And so in the core of this vesicle, it's round, are neutral lipids. And so this is where all cells store their fats. And on the surface of this lipid droplet, you'll find these proteins that exist that can regulate a variety of things. And so we're going to look at, the study is about what proteins are present on these cells. And in these non-malignant models, the lipid droplets have been investigated, and the proteins on those lipid droplets in cells like fat cells, adipocytes, or also looked in muscle cells and intestinal cells. And in those non-malignant models, lipid metabolism proteins are the most prevalent proteins. Makes sense. They're lipid droplets. They transfer fats in and out of that lipid droplet to supply the cell with energy metabolism in most cases. However, there are a variety of other proteins present on the surface of that. I wave my hand around too. So the surface of that adipocyte or that lipid droplet that are involved with other cellular regulatory processes like regulating gene expression or protein expression or inflammatory signaling. So they may actually serve completely different processes as an organelle that regulates cellular processes, and that's kind of a more recent thinking about what these lipid droplets do. But it's never been investigated in metastatic cells. So we took this on. Che, who is back there, Andolino actually did most of the work here and was a student, is now a postdoc in my lab. Excuse me. And this was a really fun collaboration with Kim Buman, who's also from my department, who is a lipid droplet expert. And we talked for years about doing this project and finally got together. And our students, Alyssa and Shay, teamed up to do the characterization of the proteome in a metastatic cell line, a breast cancer cell line, for the first time. And so the way this is done is they grow up cells. They had to grow up tons of cells to be able to get enough lipids to isolate proteins to do this. And then went through a typical isolation. They spun it so the lipids go to the top, isolate that for proteomics, and then the data analysis. I'm going to skip that. So what they found in the study was that there were over 1,500 distinct proteins found on the surface of the cytoplasmic lipid droplet, just an astounding number. And surprisingly, only 3% of those were related to lipid metabolism. So somehow or other, there were all these other proteins, but we don't know what they do in the cell. And this is very different from adipocytes or the intestinal cells. Most of their proteins are associated with lipid metabolism. So what are all these 1,500 proteins, and do they regulate metastasis in these metastatic cells? Surprisingly, a lot of the most enriched proteins were processes that occur during metastases, such as the cell-cell adhesion was the most enriched category of proteins on the surface of the droplets. And this is important because as cells progress from a normal cell to a tumor and ultimately to break away from that tumor and disperse into the bloodstream, they become single cells in most cases. And so they no longer need cell-cell adhesion. Normal cells don't survive that process. If they lack cell-cell adhesion, they die. So these metastatic cells have to overcome this stress of being separated from other cells and survive in order to metastasize. However, now that we have 1,500 proteins, it's really difficult to study because we know all these categories, but we can't toggle them on and off that lipid droplet. And what matters here is not the total amount in the cell, it matters whether they're on the lipid droplet or off. And we don't have that information to be able to toggle them on and off. So we decided to think of another way to potentially do this. So here's the lipid droplet in the cell with the proteins on it in a cell. And we in the lab know that we can change the size of this lipid droplet, our incredibly shrinking lipid droplet. We have several ways of changing the size of that lipid droplet. And when we shrink it, we know that that cell it has a reduced ability to be metastatic and to be migratory, which is something we can use in the lab, which is a critical step of metastasis. Cells change shape, they can move, which normal cells are not going to do. So that's one of our measures. So we hypothesized that maybe we could look at cell lines that have this reduced lipid droplet size in order to compare the proteins on these non-metastatic versus the metastatic lipid droplets to see if we could narrow down our search for what proteins might be regulating metastasis that either go on or off the lipid droplet. So we, with the Catherine Peachy Foundation grant, we used several different models. This is the model that I just showed you that has the 1,500 proteins. And we have several other cell lines model systems that we're using to compare with the original cell line. But I'm going to focus on this one with the FASN inhibitor, and let me tell you about that. But all of these models have decreased lipid content and are less metastatic. So we're comparing the proteome of this lipid droplet across those. But let's focus on the FASN inhibitor. And I thought I should tell you why this works. So this FASN is called fatty acid synthase, and it is a critical step to cells being able to produce fatty acids, our fat stores, and those fat stores get stored in our lipid droplet. So if you inhibit FASN, you shrink your lipid droplets in the cell, and so those are one of the model systems that we're looking at. These cells that have been inhibited, they have decreased migration and they have decreased metastasis in an animal model. So we think this is a pretty good one to compare with the original very metastatic cell line. Okay, so the results from this study, we also assessed all the proteins in the cell, so what we call the whole cell lysate. And that includes what's on the lipid droplets. And many of those proteins are similar. That's this, I don't know what you call it, gray section on the middle. So common to both cells, the metastatic and the non-metastatic, there are many proteins that are similar. But what is really interesting is the proteins that were found on the lipid droplets alone in the control or metastatic, that's in the blue arc here, there are still over 1,500 proteins that were unique to that metastatic cell line that were not found on the non-metastatic cells. And there were only 9 distinct proteins found on the non-metastatic lipid droplets. So we still have a lot of proteins to be looking at. But we decided to look for categories that might be we're most interested in proteins that will move from the cytosol to the lipid droplets, so distinct protein distribution between these two cells. And I'm going to show you one more metabolic process that occurs. One that stood out just plain as day was this concept of ferroptosis. And this is a new way that cells die. And it's with iron plus reactive oxygen species, or ROS, cause lipids to peroxidase. You think about rancid lipids, it's essentially the same thing. They oxidize, and that causes cell death. So this is one way that cells can die if they have too much ROS. But what's interesting is that metastatic cells, well, all cancer cells, but also metastatic cells, have a greatly increased rate of cellular metabolism, right? So that leads to greatly increased ROS. So these metastatic cells have to adapt in such a way that they can protect themselves from this form of cell death, because it's so important for them to survive this in order to survive the metastatic process, or to successfully metastasize to a secondary site. And so I'm going to show you another very complex, it amazes me, biology amazes me, honestly, because all the green proteins here are anti-ferroptotic in a cell. All the red proteins are promote ferroptosis. How can there be so many proteins that regulate the cellular process? Amazing. So anyways, there are all these different proteins. Greens are anti, red are promote ferroptosis. And so it was a very unique redistribution of these proteins between the non-metastatic and the metastatic cells. And I'm going to show you a little bit of that data. There actually were many other proteins that were differentially expressed. So these are the proteins that promote ferroptosis. And interestingly, the amount of that protein in the whole cell is the same, right? So this is really important. There's the same amount in the total protein, in the total cell. But if you look at what the proteins that are on the lipid droplets only, all of these proteins were either higher in the metastatic cells or only found in the metastatic cells. So this suggests that these proteins are able to move from the cytosol to the lipid droplet, and that may be involved in promoting apoptosis or ferroptosis and cell death. Sorry, I said that the wrong way. I was trying to be so careful. So if maybe those proteins are stuck to the lipid droplet, and that prevents those promoting proteins to promote ferroptosis in the cell. So maybe this is a protective mechanism for the cells. I said it right that time, Jay. So that was our hypothesis, and we tested it with a number of different assays, but I wanted to show you one of them. So we use what is Erastin, which promotes ferroptosis in the cell. So it's kind of a surrogate for reactive oxygen species. And so these blue bars are our metastatic control cells that I've showed you data on. If you treat them with Erastin, they die. But if you take the non-metastatic cells with these smaller lipid droplets and the redistribution of the proteins, they die much more when they're stimulated with ferroptosis, suggesting that yes, our metastatic cells are much more protected from ferroptosis. So we still don't know if that redistribution is really the cause for this protective effect, but that's something we're going to continue to try to figure out. Now that we know proteins that actually move from the cytosol to the lipid droplet, we may be able to identify pieces of the protein that drive them to the lipid droplet, but at this point we don't know. So just in summary, this CLD accumulation and protein localization may protect against lipid peroxidation-mediated cell death in these metastatic breast cancer cells. So that redistribution on and off the lipid droplet may have a regulatory role in the cell to promote metastasis, and maybe there are ways to target it, either to reduce the lipid droplet size or to specifically target the ability of these proteins to go on and off the lipid droplet. And I think I'm before time. So I'd like to thank, again, this was a really fun collaboration with Kim Buman to bring together two different expertise, and Alyssa, and of course Shaylin, who really did all of this work, and she can probably answer the questions much better than I. And again, thank you to Catherine Peachy for supporting this work, and I also want to welcome Anna Maria Storniolo, who is also from Peachy, who is here, and so thank you. I'm happy to answer any questions.